So what we addressed in the last lecture was that as blood travels through the glomerulus, lots of blood plasma, due to the Starling forces, namely glomerular hydrostatic pressure, lots of blood plasma leaves the glomerulus and it goes into Bowman's capsule. Okay, so now we have all of this blood plasma that exists inside of Bowman's capsule. And you think, well, where is the blood plasma going to go? Well, the blood plasma is going to travel from Bowman's capsule, and it's going to begin to enter into the rest of the nephron. If you remember, what comes after Bowman's capsule is the proximal convoluted tubules. So blood plasma will go from Bowman's capsule into the proximal convoluted tubule. And now we have to start talking about what's the point of that blood plasma traveling through the proximal convoluted tubule. And I think we know that ultimately it's for the purpose of creating what will ultimately be urine. But what does that process actually entail? So we can take a look at this and begin to get a better idea. So we've got this blood plasma that exists inside of Bowman's capsule, and the blood plasma is now beginning to travel through the beginning, let's say the next part of the nephron, which is the proximal convoluted tubule. Now, what's going to happen to that blood plasma as it travels through the proximal convoluted tubule? And the first thing that I want to show you is what happens to the efferent arteriole. Okay, remember, the efferent arteriole is the vessel that leaves Bowman's capsule. Okay. All the glomerular capillaries combine and form a vessel that then leaves Bowman's capsule, and that vessel takes blood away from Bowman's capsule. We call it the efferent arteriole, right? It's right here. Now, what is going to happen to the efferent arteriole? Well, as the efferent arteriole leaves Bowman's capsule, what you can see is that it actually then forms a capillary bed. Okay? And that capillary bed actually exists around the entire nephron. All of these capillaries are called the peritubular capillaries. The efferent arteriole actually creates the peritubular capillaries. What the peritubular capillaries are is they're a capillary bed that actually exists around the entire nephron. It's a series of capillaries that exists around the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. And this capillary bed is formed actually from the efferent arteriole. So what's the point of having these capillaries lie next to the nephron? I mean, if you look here, right, what we're talking about is the blood plasma traveling through the proximal convoluted tubule. And as you can clearly see in this image, well, these peritubular capillaries, which are formed by the efferent arteriole, these peritubular capillaries exist all around the proximal convoluted tubule and the loop of Henle and everything else too. But the point is, we've got this blood plasma that's traveling through the proximal convoluted tubule, and at the same time, we have all these peritubular capillaries that are surrounding the proximal convoluted tubule. So what does all of this mean? So number one, before we get too complicated, the blood plasma that is traveling through the proximal convoluted tubule and it's going to travel through the loop of Henle and everything else, there's a lot of different molecules and ions that exist within that fluid, okay, within that plasma. And this is by no means a comprehensive list. But this is some of what exists within that blood plasma that leaves the glomerulus, goes into Bowman's capsule, and then travels throughout the rest of the nephron. This is the blood plasma that we're talking about. It's mostly made up of water, because blood plasma is always mostly made up of water. But then also, what exists within that blood plasma that's actually inside of the nephron right now? We have things like glucose, urea, sodium, potassium, calcium, chloride, etc. There's lots of different molecules and ions that actually exist within that blood plasma that's traveling through the nephron. And the point is, a lot of this stuff that's in here, like water, glucose, sodium, our body doesn't want to actually lose that within our urine. Okay? So the blood plasma that's traveling through the nephron, there's a lot of stuff within that plasma that our body does not want to lose. We don't want to excrete this out of our body through urination. So what do we do with it? Right? All right, so let's focus on the image on the left, first of all. Imagine this as a portion of the proximal convoluted tubule. Okay? What's traveling through the proximal convoluted tubule? Blood plasma is. Blood plasma is traveling through the proximal convoluted tubule. That blood plasma has water within it, glucose, sodium, calcium, etc., all within it. Okay, and it's traveling through the proximal convoluted tubule. What is the proximal convoluted tubule? And what's the whole nephron for that matter, right? The whole nephron is just this one long continuous tube, right? So whether we're looking at the proximal convoluted tubule as we are here, or we're looking at the Lupa family, or we're looking at the distal convoluted tubule, the whole nephron is this long continuous tube that blood plasma travels through. And hopefully this image does a pretty good job of depicting that, right? That's the proximal convoluted tubule. And what's traveling through the proximal convoluted tubule is blood plasma. Okay. All right. So first things first here. Tubes in our body have to be created by something and lined by something. Okay, so if you think of a blood vessel, like a blood vessel is a tube, just like the nephron is a tube. And a blood vessel is lined by a singular layer of flat-shaped epithelial cells. It's called simple squamous epithelium. Now, the nephron is a tube as well. But the nephron is lined by a singular layer of cube-shaped epithelial cells. You can actually see that here. Okay. The nephron, whether you're talking about the proximal convoluted tubule, the Lupa family, the distal, whatever, the tube itself, the nephron is a long continuous tube, and it's lined by a singular layer of cube-shaped epithelial cells, something that we call simple cuboidal epithelium. For those of you that have had anatomy, maybe remember this. Simple cuboidal epithelium is what lines the entire nephron, this long continuous tube. Okay? So for the moment, we're not going to go any further than just saying the nephron is actually made up of and lined by a layer of simple cuboidal epithelium. And in the center, there's a lumen, a hollow opening, that blood plasma actually travels through. Okay, just keep that in mind for now. Now, what lies around the entire nephron? Well, the peritubular capillaries. These are peritubular capillaries surrounding the nephron. Like if, again, we're looking at one specific port here. This is the, I'm saying that this is the proximal convoluted tubule, right? This is right after Bowman's capsule. And these are peritubular capillaries surrounding the proximal convoluted tubule. What forms the peritubular capillaries? Well, the peritubular capillaries ultimately are formed by the efferent arteriole. The efferent arteriole leaves Bowman's capsule, and then it turns into a capillary bed that we call the peritubular capillaries. And here you can see the peritubular capillaries, in my example, surrounding the proximal convoluted tubule here. Now, I just said this But what are blood vessels lined by? The inner layer of a blood vessel, and a capillary is mostly just made up of almost nothing but this. It's lined by a layer of simple squamous epithelium. Okay? A singular layer of flat-shaped epithelial cells. That's what lines blood vessels. Okay? So what you've ultimately got here is you've got two tubes lying right next to each other. Don't you? Think about it. You've got one tube that is the actual nephron itself. In this example, the proximal convoluted tubule. That's a tube. And then lying next to the nephron, or the proximal convoluted tubule in this example, are the capillaries, which are also tubes. Okay? So what's traveling through each one of these tubes? First of all, what's traveling through the nephron, in this case the proximal convoluted tubule, is blood plasma. Okay? Blood plasma is traveling through the nephron, the proximal convoluted tubule here. And what's traveling through the capillaries? Well, what's traveling through the capillaries is blood, right? Red blood cells and blood plasma, and a lot of components of the blood plasma that actually never left the glomerulus too. So you have these two tubes lying right next to each other. You have the nephron, which is a long continuous tube that has blood plasma within it. And then you have the capillaries that are lying right next to the nephron, and the capillaries are tubes that have blood inside of them. So what's the point of this whole organization? So the purpose of going through all the anatomy of that was to be able to make sense of this image here. Okay. Right here, imagine this as the proximal convoluted tubule. Okay. These are the two walls of the proximal convoluted tubule, right here and right here. Okay. What makes up the walls of the proximal convoluted tubule? Well, a layer of simple cuboidal epithelium. Okay. Simple cuboidal epithelium is what lines the proximal convoluted tubule and the entire nephron for that matter. So in the center is what's called the lumen, and that's what blood plasma travels through. You can see the blood plasma would be traveling through the lumen of the proximal convoluted tubule and the entire nephron. Okay? So as the blood plasma travels through the lumen, the blood plasma has, of course, simple cuboidal epithelial cells, or we could just say cuboidal epithelial cells, that lie on every side of it. Okay, we're trying to make this a two-dimensional image here, but as the blood plasma is traveling through the lumen of the proximal convoluted tubule, the blood plasma is just going to be surrounded by these cube-shaped epithelial cells. Okay. And then what lies outside of the proximal convoluted tubule? Well, the peritubular capillaries do. Okay, this is supposed to be here one peritubular capillary on the right. Okay, the peritubular capillary, right? That's a capillary. That's a tube that has blood within it. Lies next to the proximal convoluted tubule. This is trying to show how they lie right next to each other. And you can see, well, okay, what is the peritubular capillary, which is again a capillary, so a blood vessel? What is it lined by? Well, it's lined by a layer of simple squamous epithelium, a layer of flat-shaped epithelial cells, which you can clearly see depicted in that image. So what this is trying to show you is it's trying to show you, whether this is the proximal convoluted tubule or somewhere else, it's trying to show you how the nephron lies next to the peritubular capillaries. And what you have traveling through the nephron, through the lumen of the nephron, is blood plasma. And what you have traveling through the lumen of the peritubular capillaries is blood. Okay. And then there's one last thing to point out here, is there's something that exists in between the nephron and the peritubular capillaries, or in this case, the proximal convoluted tubule and this capillary, and that is right here, right? This stuff in here. That's fluid. That's fluid that lies directly outside of our cells, called interstitial fluid. Interstitial fluid lies in between the nephron and the peritubular capillaries. Okay? You always have that. There's always going to be fluid that exists outside of our cells, called interstitial fluid. Now, in the case of the nephron, instead of calling it interstitial fluid, it gets called peritubular fluid. But peritubular fluid is interstitial fluid. Again, you see this a lot in anatomy and physiology where you rename things specific to the organ that's being discussed. But peritubular fluid is interstitial fluid. It's the fluid that lies directly outside of the nephron and sits in between the nephron and the peritubular capillaries. Okay, so now you hopefully have the anatomical knowledge to be able to make sense of what's about to happen here. We've got blood plasma traveling all through the proximal convoluted tubule. Okay? The blood plasma is traveling through the lumen of the proximal convoluted tubule. And the proximal convoluted tubule is lined by simple cuboidal epithelium. Now, what lies right next to the proximal convoluted tubule are all of these peritubular capillaries, which have blood inside of them. Okay? And they are lined by a layer of simple squamous epithelium. Okay, so you have the proximal convoluted tubules and the peritubular capillaries that lie right next to each other. And what exists in between the proximal convoluted tubule and the peritubular capillaries is just a little bit of fluid, which we call interstitial fluid. Okay, so what's the point of this whole organization? Well, what's within this blood plasma that's traveling through the proximal convoluted tubule? There's water, sodium, glucose, potassium. I mean, there's all kinds of stuff in there. Okay? And a lot of that stuff that's in the blood plasma that exists in the nephron, the proximal convoluted tubule right now, we want to get that stuff out of the nephron and back into our bloodstream. Another way of saying that is we want to get a lot of the good stuff that is within the blood plasma that's inside of the proximal convoluted tubule. We want to get a lot of that good stuff out of the nephron, out of the proximal convoluted tubule, and into the peritubular capillaries. We want to basically get the good stuff that's within the blood plasma inside of the proximal convoluted tubule. We want to get it out of the proximal convoluted tubule and back into our bloodstream. And what that means is into the blood within the peritubular capillaries. So this is a list that you've seen before. Okay, it's by no means a comprehensive list. It's quite abbreviated, actually. But these are some of the molecules and ions that exist within the blood plasma that's inside of the nephron. Okay. And every single one of these molecules and ions, we want to get a pretty good percentage of these molecules and ions to move from the blood plasma within the nephron back into our bloodstream. Meaning we want to get the majority of all of these molecules and ions. We want to get the majority of them, as in terms of talking about a percentage. We want to get a majority of them to move from the blood plasma that's inside of the nephron. And we want to get them to move out of the nephron and go back into the blood within the peritubular capillaries. That would bring these molecules back into our bloodstream. So you can imagine when you start talking about true renal physiology, it's incredibly extensive because you get into a conversation about how every single one of these different molecules and ions is ultimately moved from the blood plasma within the nephron back into the bloodstream within the peritubular capillaries. And some of them, the way that it's accomplished is incredibly sophisticated, and it happens at different levels of the nephron, and sometimes they'll move in opposite directions, meaning that sometimes it'll go from the nephron into the bloodstream, and then sometimes it'll go back and then back again. It's very complicated and requires an extensive amount of discussion and lecturing to get through in reality. So what all we can do is focus on a couple of these. We are mostly going to focus on water and sodium. And what I mean is I'm going to show you how water and sodium within the blood plasma that exists inside of the nephron moves from the blood plasma within the nephron back into our bloodstream. I'm going to show you how water and sodium move from the blood plasma within the nephron, out of the nephron, and into the blood within the peritubular capillaries. Again, another way of saying back into our bloodstream. Okay, so let's take a look at that. So yet again, here is blood plasma traveling through a portion of the proximal convoluted tubule. Okay, and the proximal convoluted tubule is lined by these cuboidal epithelial cells. Okay. Now, what's within this blood plasma that is traveling through the lumen of the proximal convoluted tubule? Well, there is water in there, and there's sodium in there. There's many other things in there, as you know, but we're going to focus on water, H2O, and sodium. Okay. How can we get this water and this sodium to move from the nephron, right, from being inside of the lumen of the nephron, back into our bloodstream, right, back into the peritubular capillary? That is the question here. Okay. And it's complicated, and it's going to require you to think about some things that you probably haven't in a while. Okay. These cuboidal epithelial cells that line the nephron, they're cells. Okay, every single one of them is a cell. Okay, they're cuboidal epithelial cells. What do cells have? Everything that's a cell has a plasma membrane. So this is the plasma membrane of one cuboidal epithelial cell. Every single one of these cuboidal epithelial cells has a plasma membrane. They're all cells. So the point is, if you want to get water or you want to get sodium from the lumen of the nephron out of the nephron and back into the bloodstream, you have to get water and sodium past these cuboidal epithelial cells, because the cuboidal epithelial cells are what line the nephron. Well, that seems problematic, doesn't it? Because to really get water and sodium past these cuboidal epithelial cells, what that really means is you have to get water and sodium past the plasma membrane of these epithelial cells to actually get it back out into the interstitial fluid and into the bloodstream. So how are we going to be able to do that? And we're going to only be able to do it through transport mechanisms, the things that we talked about, like, you know, within the first 2 weeks of the semester. But luckily, we've kind of been addressing them again and again throughout the semester as well, even if I haven't been saying it explicitly in the different circumstances. Let's start off with sodium. We've got sodium that exists in the lumen of the nephron. And what we want to do is we want to get sodium into the cuboidal epithelial cell. That's the first thing that we need to do. We need to move sodium from the blood plasma that exists in the lumen of the nephron, and we need to move that sodium into the cuboidal epithelial cell, one of the cuboidal epithelial cells, that is lining the nephron, in this case the proximal convoluted tubule. In order to do that, we have to get sodium past the plasma membrane of the cuboidal epithelial cell. So first things first, we would say that this is the apical membrane of the cuboidal epithelial cell. Why? Because the apical membrane is always the portion of the plasma membrane that faces the opening. Okay, the lumen is the opening. Okay? The lumen of the nephron is the opening, right? That's where blood plasma is traveling through. So the portion of the cuboidal epithelial cell, the portion of its plasma membrane that faces the opening, that faces the lumen, is called the apical side or the apical membrane. That's not the biggest thing in the world, but it is, a decently important terminology. So if you say this in, anatomically correct terms, what you really should say is you're trying to get sodium from the lumen of the nephron, or in this case, the proximal convoluted tubule. You're trying to get sodium from the lumen of the proximal convoluted tubule past the apical membrane, the apical plasma membrane of the cuboidal epithelial cell and into the cuboidal epithelial cell. And how can we do that? First things first, sodium is higher in the blood plasma that exists inside of the lumen of the proximal convoluted tubule than it is inside of the cuboidal epithelial cell lining the proximal convoluted tubule. Okay, sodium is higher in the lumen than it is inside of the epithelial cell. Okay, so it has a diffusion gradient, right? Sodium is higher in the lumen than it is inside of the epithelial cell, right? If you remember, sodium is always low inside of our cells. So inside of these cuboidal epithelial cells, the sodium concentrations inside are very low. Okay? And to start off with, the sodium concentrations within the blood plasma that's inside of the lumen of the proximal convoluted tubule, the sodium concentrations are much higher. So sodium has this diffusion gradient. It wants to move from high to low concentrations. Basically what we're saying is sodium wants to move from the lumen into the cuboidal epithelial cells. It wants to move from high to low. But in order to do that, it has to get past the plasma membrane of these cuboidal epithelial cells. And the plasma membrane, if you remember, is made up, the core of it at least, is made up of hydrocarbons. It's made up of fatty acid tails. So the whole core of this plasma membrane is nonpolar. And sodium is a positively charged ion. So the point is, sodium cannot by itself get past the plasma membrane, the apical side right now. It can't get past the apical membrane of this epithelial cell. Because the membrane, the plasma membrane of the epithelial cell is nonpolar, sodium is a positively charged ion, so there's no way for sodium to actually get past it. So then you say, well, what must be embedded into the plasma membrane of this epithelial cell? And this is true for all of them in the proximal convoluted tubule, like sodium leak channels. Sodium leak channels exist specifically on the apical plasma membrane of these cuboidal epithelial cells, the portion of the plasma membrane that faces the lumen. And these leak channels, right, they serve as a passageway for sodium ions. That actually allows sodium to travel through these leak channels. I know that's not exactly what the image is showing you, but I promise you that they're here. It allows sodium to travel through these leak channels and actually enter into the cuboidal epithelial cell. If you remember what we call this, that's great. But if not, I'll remind you, it's called diffusion through channels. Again, from the very beginning of the semester, sodium travels into these cuboidal epithelial cells through diffusion through channels. It enters in through these leak channels. This is a type of passive transport. Sodium moves from high to low without using any energy. And specifically, it does that by traveling through these sodium pores, these sodium kind of leak channels that exist. We call that diffusion through channels, a type of passive transport. That's the only way that sodium can actually move down its diffusion gradient, is if these kind of pores or channels or leak channels, whatever you want to call them, these sodium channels have to exist, because otherwise sodium can't get past the plasma membrane because it's nonpolar. So now we're getting sodium from the plasma within the lumen of the nephron into these cuboidal epithelial cells. And the way that we got sodium from the lumen into these cuboidal epithelial cells was via diffusion through channels past the apical membrane. And now the next thing that we need to do is we need to get sodium from the epithelial cell out into the interstitial fluid, which is also called the peritubular fluid. We need to now get sodium past the next portion of the cuboidal epithelial cell's plasma membrane. This side of the plasma membrane is referred to as the basolateral membrane. It's the side that doesn't face the opening, right? It faces the interstitial fluid in this example. We need to get sodium from the epithelial cell, the cuboidal epithelial cell, past the basolateral membrane and now into the interstitial fluid so that finally we can pull it into the bloodstream. But yet again, we run into a bit of a problem here, right? Because in order to do this, in order to move sodium from the epithelial cell and into the interstitial fluid, we have to get it past the plasma membrane again. We have to get it past the basolateral membrane, the portion of the plasma membrane of the epithelial cell that faces the interstitial fluid. And yet again, the plasma membrane, the core of it is nonpolar, and sodium is a positive ion. So naturally, sodium is not going to be able to get past the plasma membrane. So how do we accomplish this? Okay, so sodium is very low. This is a comparative thing. Sodium is very low in the epithelial cells, the cuboidal epithelial cells lining the nephron, specifically in this example, the proximal convoluted tubule, compared to the concentration of sodium within the interstitial or peritubular fluid. Okay, and what we want to do is we want to actually get sodium from the epithelial cell past the basolateral membrane and into the interstitial or peritubular fluid. So in order to do that, we now need to move sodium from a low to a high concentration. The sodium concentration is low within the epithelial cell, and it's higher. This is again comparative thing. It's higher comparatively in the interstitial fluid. So that would be against sodium's concentration gradient, and that's still not even considering the fact that we have to get sodium past the basolateral plasma membrane, which again is nonpolar, so sodium doesn't naturally go past it. So how are we going to be able to accomplish this? Well, we're going to need to utilize, if you remember this, active transport. Active transport is what cells use to move molecules from low to high concentrations across their membranes. So we want to move sodium from a low concentration in the epithelial cell to a high concentration in the peritubular fluid. And we need to get sodium past the basolateral plasma membrane in order to do this anyways. So what's embedded into the basolateral plasma membrane of these cuboidal epithelial cells, the side that's facing the interstitial fluid, are sodium-potassium pumps. Sodium-potassium pumps are a type of primary active transport. They utilize ATP to move sodium and potassium from low to high concentrations. Let's just focus on sodium. I don't mean to confuse you too much, although we've talked about these channels so many times throughout the course of the semester. What the sodium-potassium pumps will do is they'll take sodium from a low concentration within the cuboidal epithelial cell and pump it past the basolateral membrane into a high concentration within the interstitial or peritubular fluid, as it's called. And they utilize ATP in order to be able to do this. The sodium-potassium pumps do. That's why we call it primary active transport. And this is what finishes the process, right? This is what will get now the sodium within the epithelial cell that came in from the blood plasma within the lumen of the nephron. This is what will get that sodium from the epithelial cell past the basolateral membrane and into the interstitial or peritubular fluid, a type of active transport. We need to move sodium from a low to a high concentration, and we're going to need energy to do it, and the energy is ATP, so we call this primary active transport. And I'm not going to get into this because it would require another hour, but due to the Starling forces exist all throughout your body. I realize I only introduced them with regard to the glomerulus, but the Starling forces are what dictate the movement of blood plasma in and out of all the capillaries in your body, actually. So it's the Starling forces are much are utilized and discussed much more than in just discussing the nephron. But yeah, that is what it is. Okay, so the point I'm trying to make is actually due to the Starling forces, this sodium that's now within the peritubular or interstitial fluid get actually then pulled into the blood within the peritubular capillaries. So that's the last step in the process. Let's think about what you've actually truly accomplished here, though. So what you actually accomplished here is a process that's called reabsorption of sodium. You reabsorbed sodium. It's a term that's used in renal physiology, and what reabsorption of sodium actually means is that you were able to take sodium from the lumen of the proximal convoluted tubule. Right? Sodium exists in the blood plasma that's inside of the lumen of the proximal convoluted tubule. And you were able to take that sodium and bring it out of the proximal convoluted tubule, right? Out of the nephron and back into the bloodstream within the peritubular capillaries. You reabsorbed sodium back into the bloodstream. That's what reabsorption actually means. And why would we do this? Well, because we don't actually want to lose a whole lot of sodium within our urine. We want to pull most of that sodium that's within the nephron back into our bloodstream. That's called reabsorbing the sodium. And in order to do that, we needed to get sodium from the lumen past both the apical and basolateral plasma membrane of these cuboidal epithelial cells. We got the sodium past the apical plasma membrane via diffusion through channels, right? It's a type of passive transport. Sodium moved from high to low through sodium pores or sodium channels past this apical membrane. So we used a type of passive transport, diffusion through channels, to get the sodium from the lumen past the apical membrane and into the cuboidal epithelial cell. Then we used a type of primary active transport to bring the sodium from the cuboidal epithelial cell past the basolateral membrane and into the peritubular or interstitial fluid. We had to because sodium was low inside the cuboidal epithelial cell and higher comparatively within the peritubular or interstitial fluid. So to move sodium from high to low, we're going to need a type of active transport. We're going to need energy in order to get sodium past a membrane if we want to move it from low to high. So conveniently, we have these sodium-potassium pumps that are embedded into the basolateral membranes of our cuboidal epithelial cells. And they work to pump the sodium from low concentrations in the cuboidal epithelial cells to pass the basolateral membrane and into higher concentrations within the peritubular or interstitial fluid, so that then finally sodium, once it's within the interstitial or peritubular fluid, can be brought back into the bloodstream. So as blood plasma travels through the proximal convoluted tubule, a very large percentage of the sodium within the blood plasma inside of the proximal convoluted tubule is reabsorbed back into the bloodstream. By that I mean a lot of the sodium within the blood plasma inside of the proximal convoluted tubule is brought back into the bloodstream that exists inside of the peritubular capillaries. Again, we call that reabsorbing sodium back into the blood. So this is important because over 99% of the sodium gets reabsorbed. Okay? Meaning that there's very little sodium that ultimately ends up staying within the blood plasma inside of the nephron. There's very little sodium that will actually end up being inside of your urine. There's some. Not all sodium gets reabsorbed. Some sodium stays within the plasma inside of the nephron, and ultimately, I mean, if you follow the blood plasma throughout the loop of Henle, distal convoluted tubule, the vast majority of sodium gets reabsorbed back into the bloodstream. But some stays within the plasma, even that enters into the collecting duct. So that should tell you that, you know, when urine has been created, there is still some sodium that exists inside of the urine, but 99% of it 99% of the sodium does get reabsorbed back into your bloodstream. That's in the average person that doesn't have different types of diseases and is assuming that they're following a standard diet. If you eat large or low quantities of sodium, that can influence this too. But generally speaking, the vast majority of sodium within the blood plasma inside of the nephron gets reabsorbed. So the next thing to discuss is water. Okay, so blood plasma that travels throughout the lumen of the nephron is mostly made up of water, right? This blood plasma that is traveling throughout the nephron is mostly made up of water. That's what blood plasma is mostly made up of. And no surprise here. Most of this water that's traveling throughout the lumen of the nephron, most of this water is actually going to get reabsorbed back into your bloodstream. Water we would consider to be the good stuff. Okay? We want to reabsorb most of the water. By that I mean we want to bring most of the water inside of the lumen of the nephron back into our bloodstream. And in order to do that, we need to get water, H2O, past these cuboidal epithelial cells that line the nephron in order to get that water into the interstitial or peritubular fluid and back into the bloodstream within the peritubular capillaries. So how do we accomplish that? All right, so first of all, what happened to sodium? The sodium that was traveling through the lumen of the nephron, through the lumen of the proximal convoluted tubule, the sodium was actually brought from the lumen past these cuboidal epithelial cells, through diffusion through channels, past the apical membrane and through primary active transport, past the basolateral membrane, back into the peritubular or interstitial fluid. So over time, as the sodium is being brought from the lumen of the proximal convoluted tubule, past these cuboidal epithelial cells, and into the peritubular or interstitial fluid, what's going to happen to the concentration of sodium within the peritubular or interstitial fluid? And what's going to happen to the concentration of sodium within the plasma that's inside of the lumen of the nephron? The obvious answer is the concentration of sodium within the lumen of the proximal convoluted tubule will begin to fall. And the concentration of sodium within the peritubular or interstitial fluid will begin to rise. This only makes sense, right? Because what are we doing, at the level of the proximal convoluted tubule? We're reabsorbing a whole bunch of the sodium. We're progressively taking sodium from the blood plasma within the lumen of the proximal convoluted tubule and bringing it back into the bloodstream. And by doing that, we're bringing sodium from the lumen of the proximal convoluted tubule into the peritubular or interstitial fluid. So by consequence, as we do this progressively, the concentration of sodium within the plasma that exists inside of the lumen of the proximal convoluted tubule will fall. And the concentration of sodium within the interstitial or peritubular fluid will rise as this process happens progressively, because we just keep bringing more and more sodium from the lumen of the proximal convoluted tubule into the peritubular or interstitial fluid. So all of this relates to the reabsorption of water. So where am I going with all of this? Okay, so first of all, embedded into the plasma membranes of all of these cuboidal epithelial cells throughout the proximal convoluted tubule are aquaporins. Aquaporins, water pores, are embedded into the plasma membrane of all of these cuboidal epithelial cells that line the proximal convoluted tubule. And the aquaporins you find both within the apical plasma membrane and basolateral plasma membrane. So what does that mean? Okay, well, what are we doing to the solute concentration within the plasma that's inside of the proximal convoluted tubule? We're decreasing the solute concentration by removing sodium and actually other ions and molecules that we haven't discussed, but we're decreasing the solute concentration. So what are we doing to the water concentration? We're increasing it. And what are we doing to the peritubular or interstitial fluid? Well, we're really increasing the solute concentration, and thus we are decreasing the water concentration. As we bring sodium, as well as other molecules and ions that I haven't had time to discuss, as we bring those molecules and ions from the blood plasma within the lumen of the proximal convoluted tubule, and we bring these molecules and ions into the interstitial or peritubular fluid, what we're doing, of course, is we're decreasing the solute concentration of the blood plasma within the lumen of the proximal convoluted tubule, and thus we're driving up the water concentration. And within the peritubular or interstitial fluid, as more and more molecules and ions move into this peritubular interstitial fluid, the solute concentration just keeps rising. And as the solute concentration rises, the water concentration falls. So what's ultimately going to happen here, right? This is going to be osmosis. It's going to drive osmosis. Water always wants to move from high concentrations to low concentrations through osmosis. Where is the concentration of water high right now? The concentration of water is high within the blood plasma that's inside of the lumen of the proximal convoluted tubule, because the solute concentration is decreasing because all the solute's going into the peritubular or interstitial fluid. And what's happening to the peritubular or interstitial fluid? Well, the solute concentration is rising, so the water concentration is decreasing. The water concentration within the peritubular or interstitial fluid is low. So the water concentration is high within the blood plasma inside of the lumen of the proximal convoluted tubules, and the water concentration is low within the peritubular or interstitial fluid. So water wants to move from high to low concentrations. Water wants to move from the blood plasma within the lumen of the proximal convoluted tubule into the peritubular or interstitial fluid. Right? Water wants to move from a high concentration inside of the lumen to a low concentration inside of the peritubular or interstitial fluid. And how does it do that? Well, it just tears right through these aquaporins that exist both within the apical and basolateral membranes of these cuboidal epithelial cells. And then through osmosis, which is a type of diffusion through channels, by the way, right? A type of passive transport where molecules move from high to low through channels. Through this, water is able to move from the lumen of the proximal convoluted tubules out of the proximal convoluted tubules, meaning past these cuboidal epithelial cells and into the peritubular or interstitial fluid, where then the water can be brought back into the blood within the peritubular capillaries. It's going to be Starling forces that bring the water back in. This is how water is reabsorbed from the proximal convoluted tubules. It's through osmosis. It's through diffusion through channels. As the solute moves from the lumen into the peritubular or interstitial fluid, it decreases the solute concentration in the lumen and drives up the water concentration, simultaneously increasing the solute concentration within the peritubular fluid and decreasing the water concentration. And that creates a gradient for water to move from a high concentration in the lumen to a low concentration in the peritubular or interstitial fluid. Right? It enables osmosis to occur. And then water will move through the aquaporins embedded all throughout the apical and basolateral plasma membranes of these cuboidal epithelial cells, via osmosis and move into the peritubular or interstitial fluid.
To summarize, as blood plasma travels throughout the proximal convoluted tubule, what happens is approximately 70% of both the water and the sodium that exists within that blood plasma gets reabsorbed back into the blood within the peritubular capillaries. The objective of this lecture was to show you how sodium and water can move from the lumen of the proximal convoluted tubules past the wall of the proximal convoluted tubules into the peritubular fluid and then into the blood within the peritubular capillaries. So hopefully that made sense, but now we still have some sodium and water that exists within the nephron, right? It hasn't all been reabsorbed within the, or reabsorbed from the proximal convoluted tubule. So it's going to keep traveling now down into the loop of Henle. So in the next lecture, we're going to do a couple things. We're going to take a look at what happens as this blood plasma continues to travel through the loop of Henle. And we're also going to go backwards into the proximal convoluted tubule, and we're going to take a look at how glucose gets reabsorbed.